Good morning, students. Hope you all are doing well. Um, Due to this unexpected situation of corona outbreak, we are doing some, online lectures for you. Just take good care of your health, stay healthy, and keep studying, okay? Because your final exams will be coming soon. So there won't be any extension for final exams. So make sure you all do your study well at home. All right, so today we will be covering our Unit 3 of Immunotechnology, where we have cytokines, complement system, cell mediated effector response, hypersensitivity reaction, and inflammation. So, for now, for today's lecture, I will be covering cytokines. All right, so what are cytokines? So, we have discussed, and in our previous unit, we have seen lymphoid cells, inflammatory cells, hematopoietic cells, how they are activated, how they are proliferated, how they are interacting with each other. So, for their effective interaction, for effective interaction of lymphoid cells, inflammatory cells, and hematopoietic cells, these cytokines are mandatory. These are the one which mediate the effective interaction between these group of cells. Okay, so the complex interaction among these cells are mediated by a group of protein which are collectively designated as cytokines. All right, to denote their role in cell to cell communication. So cytokines are basically low molecular weight regulatory proteins, or it could be glycoprotein as well, which are secreted by white blood cells and various other types of cells in the body in response to a number of stimuli. So, cytokines bind to specific receptors on the membrane of target cell, triggering signal transduction pathway that ultimately alter gene expression in target cell. So here in this picture you can see this is the inducing stimulus. Okay, this is the cytokine gene. This is the cell which is going to produce the cytokine after a stimulus. Once the cytokines are produced, they bind to their specific receptors. There are small receptors present on the cell membrane where these cytokines bind, okay, specifically to their receptors, and that way they facilitate a target cell to release the effective response. So, in general, the cytokines and their receptor exhibit very high affinity with each other. All right. Okay. So, a particular cytokine may bind to receptor on the membrane of same cell that secreted it exerted autocrine action. Okay. Like this one. Okay. So, what happens that a cell which is secreting a cytokine is eventually going to be the effector cell as well. So once this cell gets the stimulus, it is going to release the cytokine, and then these cytokines are going and binding on the receptors present on the same cell which is producing it. And then this type of signaling is called autocrine. Okay. Then it may bind to receptors on target cell in close proximity to the producer cell exerting paracrine action. So in this case, as you can see, if a cell is producing cytokines because of a stimulus, and then if it is going and affecting a nearby cell which has a receptor for this particular cytokine, so in each case this cytokine is different, this cytokine is different, these receptors are different, these receptors are different, and they are highly specific. So if this one is producing cytokine in paracrine signaling, okay, and then it is affecting a nearby cell, then it is called paracrine signaling. Okay, then last one is endocrine. In endocrine, what happens is a cell produces the cytokine because of a stimulus, then this cytokine goes through circulatory system and affects the other cells which are distant from this cell. Okay, and then these distant cell, they have the specific receptors for these cytokines. All right, now let us see what are certain properties of cytokines. Okay, So the main attributes exhibited by cytokines are pleiotropism, redundancy, synergism, anagonism, and cascade induction, which permits them to regulate cellular activity in coordinated interactive way. So let us see all these properties one by one, what it actually means. Pleiotropic action. Cytokine may have different biological effect on different target cell. Means, let us see, this is activated Th cell, okay? You already know that T helper cells are there, okay, and they are activated, and then they release this cytokine IL-4. Once these cytokines are produced, if they are going to affect a specific site on B cell, it will initiate three functions, that is activation of B cell, proliferation of B cell, and differentiation of B cell. However, if this IL-4 is going to affect thymocytes and mast cell, it will only initiate the proliferation. So depending on the cell type, the function is different. So these three cells have different function depending on their cell type. However, as you can see, the cytokine IL-4 is similar for all those. So same cytokine can induce different function depending on the cell type. This is called as pleiotropism. Okay. Next is redundancy. Literal meaning of redundancy is more than one cytokine are producing the same function. Okay, there is no need of using those three cytokines, but due to uh, different stimulus, all these three cytokines will be produced in different cell types, but these three cytokines will eventually lead to the same kind of function. That is why it is called redundancy. Two or more cytokines that mediate similar function are said to be redundant. So this is the cell activated, again T helper cell, and then this is going to induce uh, by a stimulus to produce these three different kinds of cytokines, and these three cytokines will eventually do and perform the proliferation of B cell. Okay, then next is synergism. Okay, synergy. Okay, combine effect. Let us see. So synergism is cytokine synergism occurs when the combined effect of two cytokines on cellular activity is greater than additive effects of individual cytokines. Okay, the way it works is if you see the individual effect of IL-4 on this particular cell, that is B cell, and individual effect of IL-5 on the same cell, B cell, and then you add it, the effect is less than when these two cytokines are combined together and then going to act upon this cell. So the effect of the combining these two cytokines on the B cell is more than individual effects of these two cytokines and then checking their effect and combining it. Okay, so that is called synergism. Then antagonism. The effect of one cytokine inhibit the offset of effect of other cytokines. All right, so it's very clear here in the picture. This particular cytokine, that is IL-4, is going to initiate the function. Okay, it is going to go in a forward direction, but then IFN gamma is a cytokine which is again released by the cell, which is going to inhibit the function. Then next is the final one is cascade induction. Cascade. A series of event is known as cascade. That is the cascade. Okay, occurs when the action of one cytokine on target cell induces that cell to produce one or more other cytokines, which in turn may induce other target cell to produce other cytokines. Meaning, this particular cell is getting induced and producing one particular cytokine, and then this particular cytokine is inducing other cells, that is macrophages, to produce other cytokine. Then this particular cytokine is again inducing a group of cells and producing multiple cytokines. Okay, so this is called cascade induction. These are the main properties of cytokine. Okay, the term cytokine consist of, sometimes you may hear lymphokine, cytokine related to lymphocyte, okay, monokine secreted by monocytes or macrophages, interleukins, and so cytokine term has four different, sub cytokines. You can say, depending on the cells which are secreting it, we have divided into four classes. First one is lymphokines, secreted by lymphocytes; monokines, which are secreted by monocyte or macro and or macrophages; interleukins, cytokines which are secreted by some leukocytes and act upon other leukocytes; then chemokines, a group of low molecular weight cytokines that affect chemotaxis and other aspects of leukocyte behavior. I am sure you already are aware of chemotaxis. Stimulus, uh, a chemical stimulus can lead to movement of an organism. Okay, so in this case, a chemical stimulus, which can be inflammation due to allergy or any kind of injury, a movement of lymphocytes, leukocytes. Okay, so depending on the stimulus, how the leukocytes are moving, that is called chemotaxis in terms of uh, chemokines. All right. These terms sometimes are misleading because secretion of many lymphokines and monokines is not limited to lymphocytes and monocytes, as these terms imply but extend to broad spectrum of cell and cell types. So these terms in general we use cytokines because although we are saying lymphokines or monokines, but the secretion is not limited, okay, to lymphocytes or monocytes. So that's why we use the more general term, and that is cytokine happens. Then next is, in case of lymphoid and myeloid cancer, abnormally high level of IL-6 are secreted by cardiac myxoma cells, a benign heart tumor. In myeloma cell, IL-6 appears to operate an autocrine manner to stimulate cell proliferation. Okay, try to recall the autocrine we have studied earlier. So IL-6, the cell which is getting the stimulus and producing IL-6, is eventually going to be the effector cell for IL-6. And by doing that, it is going to proliferate into more cells. And that is how this myeloma is occurring. So when the monoclonal antibodies to these IL-6 are added to in vitro cultures, their growth was inhibited. Okay, so that showed that actually IL-6 are the one which are the cytokines which are working in autocrine signaling way and producing more cells. Uh, So I'm sure you already are aware of monoclonal antibody. Monoclonal antibodies are very highly specific antibodies which are made against a specific antigen. And in this case, it is made against IL-6 to see whether IL-6 is the actually uh, the molecule, the cytokine, which is affecting the cell proliferation, and that's what they found. Okay, so in addition, transgenic mice that express high level of IL-6 have been found to exhibit massive fatal plasma cell proliferation called plasmacytosis. Although these plasma cells are not malignant, the high rate of plasma cell proliferation possibly contributes to development of cancer. The last one is Chagas disease. So the earlier the two diseases you saw is about the overexpression of cytokine. This one is underexpression. The protozoan Trypanosoma cruzi is a causative agent of Chagas disease, which is characterized by severe immune suppression. This is characterized by dramatic reduction in the expression of 55 kilodalton subunit of IL-2 receptor. So because of the underexpression of this particular receptor, this Chagas disease is being caused. Okay, that's all for today. Tomorrow we are going to be discussing about complement system. All right, so again, please keep studying. Okay, take care. Bye bye.